This season, we celebrate 20 years as Los Angeles' premier museum after-hours experience, where exclusive nighttime access to NHM brings music, science, and culture together for an engaging evening of conversation and community. This season, we are proud to present our discussion series in collaboration with the Planetary Program at the Berggruen Institute. Tonight, we launch Life from Our Guts to the Galaxy, Rethinking the Living World, and we open with Life at the Human Scale. I want to give some special thanks to our event sponsor, the Berggruen Institute. More on that in a little bit. And our media sponsor, KCRW, and our event partners in progress for helping make this series a success. And now, with a few more words from the Berggruen Institute, please welcome Associate Director of the Planetary Program at the Berggruen Institute, Jonathan Blake.
Thank you. Good evening. I'm Jonathan Blake, the Director of the Planetary Program at the Berggruen Institute, which is an independent research center headquartered here in Los Angeles that's dedicated to the big questions at the intersection of science, philosophy, and governance. At the Berggruen Institute, we believe that nothing is more powerful than an idea whose time has come. And so we're delighted to be collaborating with the Natural History Museum on this mini festival of ideas. Through these 4 First Friday celebrations, we'll be looking at life in new ways, asking new questions, and looking at things from new angles. In particular, we're going to be looking at life on Earth, and in doing so, we'll adopt what we call a planetary perspective, which is a perspective that transcends boundaries, beyond countries, beyond the human-non-human divide, and beyond ways of knowing. So such a perspective, we believe, inspires us to think of the planet as a whole, and takes the question of life in a new light. So from this angle, the distinctions that typically draw and the social cultural divides that divide us, like those between nations and peoples, fall away. In this way, we'll peek at an emerging and evolving landscape of life on this planet and possibly beyond. So we're excited to go on this journey with you all as we consider and reconsider what it means to be alive.
Thank you, Jonathan. The Berggruen Institute has a table in our grand foyer, so make sure you check them out after the talk today. But without any further ado, now on behalf of NHM, it is my pleasure to introduce our host and moderator for the First Fridays this season, driven by a mission to redefine where science storytelling belongs and in belief in the power of science to connect people and communities. Please give a big warm hand to neuroscientist and science communicator, Dr. Yewande Pearse.
Hello, everyone, and welcome to First Fridays, 20 years. That's incredible. Thank you for the warm welcome, Marisol. Super excited for our season this year. Before we jump into the introduction of tonight's discussion, look out for this pamphlet. This beautiful pamphlet will be available at the table. It's produced by the Berggruen Institute, and will give you an overview of the season's discussion. So please make sure that you pick up one of those. But this evening's discussion is about life at the human scale, but not in the way that you might think. We're going to explore the trillions of microbes living inside us right now. Invisible partners that shape everything from how we digest our food to how we feel and think. But we'll also zoom out to take a look at these relationships across mammals and ecosystems. Tonight, we'll be asking the question, where does an individual organism begin? Or where does an individual organism end and its microbial ecosystem begin? We'll discover how these relationships reshape our understanding of identity and what this means for health, behavior, and the planet's future. By the end, you might see yourself and the natural world in an entirely different way. So I'm super excited to welcome our guests for this evening. Please welcome, first of all, Dr. Kayce Bell. Dr. Bell studies the diversity and distribution of small mammals, their parasites, and sometimes the microbes in both. Natural history collections play a vital role in her research to understand how history, landscapes, and interactions among different species shape the diversity of life that we see today. Welcome, Kayce. And our second guest, Dr. Jack Gilbert. Dr. Gilbert works at University of California, San Diego as a professor in pediatrics and Scripps Institution of Oceanography, the Associate Vice Chancellor for Marine Science and Director of the Soil Health Center. He has authored more than 450 peer-reviewed publications on microbial ecology, and he's the president of Applied Microbiology International, a fellow of the American Academy of Microbiology, and co-chair for the new IUCN Microbial Conservation Specialist Group. His research spans human and environmental microbial health with a special focus on translation. Welcome, both of you. So there's a lot to talk about, so we'll dive right in with the big picture. So I've been doing a little bit of research, so I'll do my little thing first. Over a century of biology has focused on the individual organism, but 50% of cells in your body aren't genetically you, they're actually microbial. Your gut alone contains over 150 bacterial species with a collective genome 150 times larger than your human genome. So that begs the question, where does you end and your ecosystem begin? And what do other species teach us about what it really means to be an individual? I'd love to start with you, Jack.
So it's a very big question. I'll start by...
big, we're going to narrow
and by the
down.
way, she's only choosing me because I'm English, and so she can understand the accent.
Bias.
We'll skip over the American, we'll come back to her in a bit.
Sorry, Kayce.
From my perspective, we don't have a beginning and an end, right? That sounds very existential, but it's quite accurate. Each one of you right now is releasing around 30 million bacterial cells into your immediate vicinity every hour. So the two of you sitting next to each other are actually stimulating each other's immune systems with the microbes that you're bleeding out all over the place, right? And
it's very romantic.
I know, it's very romantic. I love that you say it's romantic. That's awesome. Because some of those interactions can actually stimulate emotional reactions, right? So that relationship that we have, the microbes within us, the microbes that we expel into the world, and how they affect the world around us, actually shapes our physiology, our health, our neurological health, our well-being, our sense of self, in quite profound ways that we're only really just starting to understand.
That's fascinating. What a way to start. So it is a big question. Kayce, over to you. I'd love to hear what your perspective is on that question.
Thanks. Yeah, so I definitely think about this in mammals that aren't humans most of the time, but I also like to think about it in humans. And yeah, same, right? There are a lot of microbes around, and they're not just inside of us and on our skin. I also think a lot about the parasites that live in and on us and their role in impacting microbial communities and us as an ecosystem. So individuals, I like to think of them as their own little community, and that involves everything from the microbes all the way up to the parasites and the other things that we can be interacting with at all times.
And we're going to dive into some parasite talk later in the discussion, because I like to think of them now that I have the privilege of knowing more about them through speaking to you as like the underdog, and I think that they're really fascinating. Jack, I'd like to touch on one of my big questions. When I think about this, you painted a really engaging picture of these microbes kind of going back and forth between us. In my mind, the concept of our microbiome being at all stable is mind-blowing. Could you kind of clear that up for us? So how stable is our microbiome? It influences us and who we are, but how much does that change? How dynamic is it?
Yeah, well, it changes quite dramatically throughout life. Let's start there, right? So when you're born, you're actually in this wonderful little yolky egg sac inside your mother, the amniotic sac, and that's a pretty impermeable barrier. There's a lot of communication going from your mother to you as a fetal agent, and as you are developing, that relationship with your mother is actually intimated from the outside. So whatever she's experiencing, are there any pregnant people in the house tonight? Whatever you're experiencing, your fetus is also experiencing in an immunological way, and so that microbial interaction you're having with your fellow seat partner is also relating to you, and they're being passed on to your child. But there's no bacteria viruses, fungi in the amniotic sac. But once that ruptures, it changes quite dramatically. And the first big dose of microbes that you get really come from your mother's gastrointestinal tract. Anyone that's ever been there at a birth, and I know you two have.
I've been there.
Viscerally. And you'll notice that there's often quite a lot of physical interaction between the baby and the gastrointestinal tract, but that's good. That's actually pumping good bacteria that the mother has into the baby's body, helping the baby to develop a microbial symbiosis, which all of our ancestors, going back through our mammal lineage and then all the way back into our reptile and amphibian lineage, has always had. We can't exist in this world without managing and controlling that microbial exposure that we have. And there are literally trillions of microbes we're exposed to every day. And those interactions that we have shape not just our bodies, the development of our bodies, our immune systems, our metabolic systems, our neurological systems, but also the very ecological structure of that microbiome, that microbial ecosystem living inside us, on our skin, in our urogenital tract, in our lungs, in our eyes. And I'm not going to deal with it on the face mites that live on your skin. We'll get there later. So they are constantly permeating us. How do we deal with this? And what does it mean to be stable? We don't. We develop something called an immune system. You may have heard of it. A lot of people like to think of the immune system as an army. It's really not. It's much more like a national park warden or a gardener, right? It's there to manage this wonderful microbial ecosystem that we have access to, and to use it to produce things which we can then consume, much like a gardener grows the carrots and cabbages, right? And so that relationship changes throughout your life, through birth, through the first stages of your development, into puberty, and then into your second puberty when you get to 55. Yeah, I know. And it changes, and it's dynamic, and it's fluctuating as your body fluctuates. As you adapt, so does your microbiome. So it's not a constant thing. Interestingly, though, we do tend to develop a very similar ecosystem that's profoundly us. So the three of us, so I could sequence, this is another fun fact, if I took a swab and I swabbed the chairs that we're sitting on right now, I could identify each of the people that have been sitting on these chairs. Right? I could do the same for every one of you. I can neither confirm or deny whether the CIA is using this technology right now to track you.
Say less.
But it could be a tool. It could be a fingerprint. It's a fingerprint of you. It may change dynamically over time, but it's much like the Yellowstone National Park would change over the seasons, right? It's always going to be that ecosystem. It's just fluctuating, dynamically pulsing as you survive and thrive throughout your life.
And you've got, last year I think you published some research in the baboon population, which exemplifies that tremendously. And because I have just been pregnant, my mind was blown. So I'd love for you to sort of do the cliff notes of what you found, because it's absolutely fascinating.
Okay. No, yeah, I'll give you the cliff notes because it's fun, right? We went in and we looked at baboons. We had almost 300 baboons over a period of 10 years. And we sent graduate students and postdocs out into the field and made them individually, this is what we do, right? Make them individually collect the poops that any baboon had deposited and dutifully put it in a tube and label it as to which baboon it came from. And then we brought that back into the lab, sequenced all the genetic information in there, and yielded a picture of not just the evolution of the microbes in the individual baboons, but the social sharing of those microbes. And what we found was quite profound, that the bacteria that the baboons were sharing through grooming and physical interaction with each other were beneficial for their health. They helped the baboons to digest their food in quite specific ways. They helped the baboons to regulate their immune response. And if you were a social baboon, you interacted very carefully with your other ape friends, you would get more of those beneficial bacteria, much like a probiotic. And this is why we think social interactions between humans are so important. Think about why you evolved kissing and hugging and, well, I'm English, so we don't do that, right?
Absolutely not.
But for you Americans and your ebullience, it's wonderful to see all the physical dynamic interactions. And I've watched Bridgerton as well. But these relationships we think are really profound. We may have evolved these social dynamics to help us share beneficial bacteria. And then therefore, we're really just the puppets of our microbial masters.
Isn't that mind-blowing? I want to stick on evolution because I feel like Dr. Bell, you research how parasites can actually influence our evolution, which is another way of thinking about it. So rather than this like dynamic but in equilibrium idea of how our microbiome shifts, actually the shaping of our microbiome is something that we can look to chipmunks and their parasites too.
Yeah. So my work has been on looking at how mammals, and specifically chipmunks mostly because they're the best, have changed over time and how they've come to be the species that we see today. Because alongside them, as they have been evolving, they have these other parasites. So I specifically study the sucking lice, which are related to the lice that humans get, the head lice, and then also their intestinal roundworms that they get. And trying to look at how, so when we have something that speciates, it turns into a new species of chipmunk, what happens to the parasites? And it's really great to learn about the forces that are changing these. And so similar to the microbiomes, it's actually none of this is like stable or predictable. So these things can change over time. Excuse me, they're not predictable in the same way as like every time we get a new species of chipmunk, we don't always get a new species of parasite. And this has implications for how like these hosts' immune systems are evolving and which parasites they are used to. And sometimes being with kind of your partner that you've been with for a long time in evolution can be really beneficial because it means that there's a constant struggle between the host chipmunk and the parasitic pinworm or whatever the example is, where the parasite wants to live and the host wants to live and the host doesn't want to get sick and the parasite wants to reproduce. So there's this kind of like constant back and forth struggle for them to evolve and survive and pass on their genes, which is the basis of how we're all here today. And that can have an impact on how the immune system or how the diversity of these animals end up in the end, because the parasites just trying to live and the hosts are trying to get by with fewer of them. And that's usually what happens in the end, is they often come to this kind of equilibrium place where the chipmunk can have some pinworms that don't make it too sick, but the pinworms are able to still survive. And they're just kind of in this constant back and forth struggle.
That's so, you've described that kind of like it's co-evolution that you're talking about there. And it's funny that you mentioned the immune system, but you wouldn't have an how can you have an immune system without it having something to respond to? So I really kind of like that reframing of that relationship between parasites and organisms. We've been talking about the complexity. I think we can agree that we're ecosystems, not individuals in the ways that we thought already. But of course, ecosystems involve relationships, so we need to think about how those organisms interact with each other and their hosts. So Kayce, I'll stick with you for now, but it seems that the relationship between microbes and their hosts is rarely straightforward. So I've read about it being kind of like microbial politics being at play. So we often talk about mutualism, which would be win-win, where both sides benefit, and then parasitism is one organism benefiting at the other's expense. But in reality, the same microbe can be helpful in one situation, not so helpful in another. So really, how do you unpack that kind of gray area?
It's hard.
Yeah.
So
in conclusion, it is difficult.
It's with a lot of species interactions, and so like this, we'll start with the mutualism and parasitism thing. It's actually more of a spectrum, right? It's not very clear-cut that there are things that are always mutualists and there are things that are always parasites. So we're taught that by definition, a parasite has to cause harm. There has to be a cost, that the host is being harmed and it is not deriving a benefit. But that's probably not always the case. There are probably a lot of the time where the host is getting some kind of an indirect benefit, such as training its immune system or something like that. And so it's this spectrum. And what's really exciting about studying this spectrum and how things can go from being like a mutualist to a parasite or from a parasite to a mutualist is that there's this just wide span of things that can happen in between. And to not try to get too far sidetracked, but one of the things that I think about this too is like just a really straightforward example of this is our mitochondria, right? That's actually just like where we just like engulfed another bacteria and we're like, hey, let's take your functions on to help us work better. And so there are a lot of examples in nature now of studying this transition from parasitism to mutualism and how it's a process that we can track and we can see it happening. And we can also go back in time and look and see like everything that this thing is related to used to be a parasite. But now we know that it is a beneficial organism to have. And one really relatively recently well-studied example of that is actually lice. So sucking lice and chewing lice, they actually don't have the ability to make some of their vitamins. And so they've just co-opted bacteria, and they've said, I can't make B vitamins anymore. My genome is small, and I don't make B vitamins, but I've gotten these bacteria inside of me now that will make my B vitamins for me. So it's definitely a spectrum.
That's a great example. I'm sure there are lots that track to humans as well. I cannot remember the details. Maybe you know a little bit more than me, but the ARC protein, which is actually viral, to do with memory. So even memory formation could be influenced, but that's a whole nother topic. So in studying this, when I was sort of trying to get my head around it, I understand museums collecting specimens to study the organism, but the parasites, how do you actually go about that?
Lots of different ways. So, parasites are the ectoparasites are the things that live on the outside, like fleas and lice and ticks and mites. And then on the inside, we have the endoparasites, so the roundworms and the tapeworms and things like that. And, when we have a recently collected specimen, we can get all of those things. But part of my research has actually been taking advantage of specimens that have been in collections for hundreds of years, well, up to 100 years, a little more, when the ectoparasites they can often remain on these museum specimens. So when this chipmunk died in 1930, it had lice on it, and the lice actually stayed on that specimen through time, and I have now gone back and been able to comb the lice off of the chipmunk and actually look at the genome of the louse that was on the chipmunk before we knew what DNA was.
I love science. That's amazing. Talking about microbial politics metaphorically actually leads quite nicely into real-world politics, which I think we should take a moment to talk about. Jack, you're involved in the Microbes and Social Equity Working Group, which examines how microbiology intersects with issues of equity and justice. Does amazing work. Could you talk a little bit about that work and why understanding soil and oceanography, both research focuses in your lab, are just as important as understanding our own microbial biology?
Yeah, that's a big question. So we got another hour, right?
Yes, 2 hours.
Just keep on,
so,
2 hours.
yeah.
Great. Back in the day. So we profoundly understand ourselves as a social species, like I was saying, and how we socially interact with the world and with the people around us, with the governments that manage our expectations of our quality of life in different environments. We try to understand what role or how microbes might play a role in mediating those relationships. You can think of it like this. There's a wonderful study, very, very well-known study. Back in the 1950s, a government in the Middle East was trying to eradicate a species of infectious disease called Bordetella. And by doing this, to try and get rid of this particular pathogen, they had to go out and cull wild animals, wild goats, wild sheep, and to try and reduce the burden that would come from these populations. However, they didn't really do this in a way that managed the expectations of some of the Bedouin tribes living in the area. And so the Bedouin tribes would take their sheep and hide them, worried that the authorities were going to come along and shoot their herds. And where do you hide your sheep? You hide them in your house. And as a result, we saw a massive disease outbreak in the Bedouin populations. So this is a great example of how a political decision, a meaningfully beneficial one, that was aimed to try and improve disease burden in a population, ended up causing a more serious threat to a group of people who weren't necessarily engaged in the socially governed program of that community. And we found hundreds of examples, similar examples, where socially, politically derived scenarios lead to harm or benefit through public interaction, through government laws, engagement in social practices. So we're very, very excited to try and understand these processes, and then to try and put microbes and our understanding of disease progression, social transfer of disease, disease for etiology from animals, bats maybe, or lice or chipmunks, put that into the legal practices and the legal frameworks by which we make our laws, through which we make our decisions in how to run our societies, so that we can significantly reduce the social inequity that may occur as a result of a decision that a group of people have made to manage their environment. And I think this is a profound role. I'm a microbial centrist, right? I believe microbes are the center of everything. Yeah, as you pointed out, mitochondria,
They very well
right?
might be, yeah.
Chloroplasts are bacteria, mitochondria are bacteria, ancient ones be that. So we are all microbes originally, and to microbes we shall return. I have a religion. You can pick up my pamphlets at the end. But this, to me, is really important, right? Can we use all this information, this knowledge, to try and make real change in our society? Not only to improve our society, improve the well-being of everybody, but maybe even make a buck on the side, right? There's opportunities for microbial technologies that can improve our economic models, that can create new industries, new businesses, and therefore support local economies in a fashion which can provide local communities that would normally be left out of a large economic model the opportunity to engage in that practice. So I think microbes could save the world.
I'm sold, I think.
Yeah.
Actually. I love the connective tissue between those two things. And we're talking about like little microbes and then these big picture ideas, which is the whole point of this discussion series. And I think that really is a great example of how that works. And I know, Kayce, you also have published a paper on conserving parasites, which ironically came out in 2020 when it was the pandemic. So I'm sure no one was really thinking about wanting to save parasites, but I guess that is relevant because it also feeds into this idea of parasites having an impact on our environment and the downward effects of that. So I'd love to hear like your thoughts on the relevance.
Yeah, it's similar to what Jack was saying about the bacteria. Is like we need to think of them more as part of like a holistic approach to an animal or a population or a species, and that parasites are they're part of the community that is this individual or this species. And when you remove a parasite from a population, you actually can't always anticipate the consequences of that. So a lot of the times people would say, by definition, parasites are harmful, and so taking them away must be a good thing. But what that can often do is leave room for something that maybe this species hasn't co-evolved with. So its immune system is not prepared to fight off this new thing that is going to come in because it doesn't have the parasites that it has co-evolved with, and it's ready to fight those off. The other part of that is that we can also kind of think of parasites as like mini predators, right? They can play a really important role in things like population control. And so we could, if you remove a parasite from an ecosystem, there could be some kind of organism. There's some species whose populations just explode, and that has cascading impacts that we can't anticipate because we have no idea what these types of ecosystem disruptions can cause. And the other part of it, why we really actually are promoting, we actually recently formed, I've been invited to participate in something somebody else did, the IUCN Parasite Conservation Group, because they're not actually being assessed as these groups that should be conserved. And so we're trying to work on that. But the other part of it is thinking of them as indicators of ecosystem health, because a lot of parasites require multiple hosts to complete their life cycle. So they don't just live in a mouse, they also live in a snail, and they have to go from a duck to a snail to a mouse to a duck or something like that. So there's lots of opportunities to actually know that if there's a parasite in this ecosystem, all of the other hosts are also there.
That's fascinating. That's really, I mean, the framing, once again, thinking about it differently. I don't think you would anticipate that parasites could have such impact. So zooming out, we've just been talking about big picture ideas, evolution, climate change, and global challenges. I want to zoom back in now to something much more personal, which is what's happening inside of our own bodies. And as a neuroscientist, I'm really interested in this, particularly our brains. I think this is really juicy territory. So interest in the gut-brain axis has exploded in recent years, the connection between our microbiomes and our brains. And so it's brought neuroscience and ecology into closer conversation than ever before. So, Jack, could you explain why the microbiome has increasingly become more and more relevant to our mental health and kind of what you're finding from your research?
I would argue it's always been relevant to our mental health. It's just neuroscientists like yourself are waking up. But to the microbial overlords. It's been fascinating. It's been a fascinating journey. My family has a history of depression. I definitely have ADHD. I have no ability to focus or pay attention to anything for very much longer than I can. So I think I've always had a profound interest in trying to understand the role that the microbes inside us may play in mediating neurological disorders, neurological diseases, and different neurotypes. The opportunity for us all to be different and to have a different perspective on the world. And of course, because I'm a microbial evangelist, I believe that the microbes must be playing a role in that. And so we've spent approximately 15 years trying to develop data that supports that conclusion. And what we found is quite profound. I believe so, that microbes play a fundamental role in shaping the health of your brain. Not just how your brain prunes neurons over time and shapes, therefore, memory and intellect, but also how it can damage through inflammation, through changes in neurotransmitter levels in the brain, which have profound impacts upon how we feel emotionally. We now know that we can certain people who are suffering from major depressive disorder are missing certain species of bacteria in their intestine that can be easily wiped out by eating the wrong types of foods, what I refer to as the standard American diet, or the SAD diet.
I think we're outnumbered. I think you need to be careful.
I don't know. You can attack me afterwards. But a high saturated fat, high sugar diet can often lead to some of these species being eradicated, which can leave us at risk of developing depression, anxiety, sociological stress results, which we've seen in our population significantly increasing. We found in animal studies that if we add these bacteria back into models of animals that are showing some symptoms of depression, that we can return or alleviate those symptoms in the same level as common chemical antidepressants like ketamine, right? So think about that. Ketamine and many of the antidepressants, the SSRIs, serotonin reuptake inhibitors that we use quite frequently to treat depression, do have side effects. They also do have a profound or a profound lack of functionality in many, many different people, right? They only are effective in certain people. So developing an entire new suite of potentially probiotic antidepressants could be incredibly valuable. Rebalancing your ecosystem, like putting the parasites back in the right place in that loop, putting the microbes back in the right place in the loop of your body and your brain, could play a profound role in helping to alleviate one of the major causes of neurological distress in our country.
And this is a huge area of, I would call it emerging, but as you say, or you've been kind of advocating for this for years. But I just wanted to take a step back and just think about sort of our role in, ironically, influencing the impact of microbes on our cells. So you wrote a book, Dirt is Good, about this idea of dewilding the human microbiome, and you've mentioned a couple of things: antibiotics, C-sections, processed foods. These all influence that. So so this would be a pathway to regaining some of that equilibrium, but also with fecal matter transplants, you're saying that this would be a way of actually curing disease as well.
So adding microbes back into our world can be beneficial. We have a great study where we take mothers and take their poop. And like I talked about with birth, but we do it literally. We take their poop, slurry it up in a blender, make a little cocktail out of it. It's yummy, and I can sell that outside. And put it back into the baby's intestine. Literally, your mother giving you the poop. I could say it in a different way, but I can't, can I?
It's perfect.
I can be good, right? I'm just gonna check that I can. She's like, yes, I can. I'm having a conversation with her now. ADHD, sorry. Yeah, so we can put the bacteria back in, and they can have profound effects upon the development of our immune system, upon our neurological health and well-being. But we can also take people and put them back into the outside world. So we have a study going on right now with a colleague of mine, Amber Fife Johnson, up in Washington State, where we are rewilding a child's experience, taking children preschool, so these are 2 to 3 year old kids, you may have one of those, and putting them into what we refer to as a forest school. They learn all the same tools and techniques and approaches that they would get in a standard preschool, but they have also the opportunity to interact with a forest environment, a wild forest environment. And that seems to have a profound impact upon their immunological and neurological development. We see children with lower levels of systemic inflammation. They don't have as much cortisol pumping around their bloodstream. They have a lower chance of developing obesity and metabolic diseases by the age of 5. They even tend to have higher school readiness scores when they go to kindergarten than children that grow up and are going into preschools in an urban environment. So there is a profound opportunity to do that. The Finnish, I won't finish, I will finish. The Finnish did it really well. They took the forest soil and brought it into the urban clinics, into the urban preschools, and let the kids just muck around in these tubs of dirt that they brought in from the forests of northern Finland, and it had the same effect. So you don't necessarily have to take the kids to the forest, you can take the forest to the kids and get a similar benefit. Why is that important? You're just stimulating. Your skin is a monstrously big immune system responder. It's like the NSA, right? It's got thousands of little motives. High five me, bam. We just exchanged microbes and we exchanged an immunological signature. Now, my immune system and your immune system are recognizing that physical interaction, not just from physical touch, but from a microbiological perspective, like I said earlier, right? So just getting your kids out there, letting them physically interact with a furry pet can reduce the chances of developing atopy and allergic disease by 13 to 14%. Letting them grow up in a farming environment or have an opportunity to interact with a farmyard environment can reduce it by 50%. Having children physically interact with dirt, and by dirt, for all of the soil scientists out there, I deeply apologize, I mean soil, healthy soil, can have a profound impact upon their health and development. And so we have a real opportunity to do something powerful to shape the health of our children and of our community.
And I love this because it's all actionable. This is all within our control, and there's a full spectrum from just generally being healthier to actually curing disease. But I wanted to take this moment, just for my own benefit, really. It is kind of hard to navigate. So I'm like, with the kimchi, yes, the kimchi, but then I'm a sucker for those probiotic sodas because it says it's good for my microbiome. Is there anything you could debunk that isn't, in terms of probiotics? Is there anything, just maybe one thing where this doesn't necessarily apply? You mentioned dirt, which is interesting because I know we were, especially with toddlers, you're sanitizing everything. So soil, dirt, maybe you could focus on that as a differentiation.
Soil, dirt. So soil, most of our soils are being severely degraded around the world due to changes in climate, due to intensive agriculture, our use of fertilizers, herbicides, pesticides, and various other features, monocultures, which are just wiping them out. We are trying to repopulate those soils, just the way we repopulate our bodies with fecal. Thank you very much. Here all night. With our fecal microbiome transplants, we're giving soil transplants back into soil environments, and that can make those soils profoundly healthy. What I think is very, very interesting is our ability to take that kind of paradigm and translate it into human health outcomes and merge those two processes, whereby a healthy environment can shape a healthy body, and you can then bring a child up in an environment which is healthy, and their body will be healthy too.
Thanks for that. And as we just end this discussion, I'd like to come back to you, Kayce. I think as we focus on connecting with not just ourselves, but with the environment around us, I know that you've learned a lot from your research in other mammals about how context matters. So I wondered if you could add a few words about kind of as we think of the power of the microbiome and all of these interventions, how we should think about that in the context of your work.
Yeah, thank you. So the context, it's kind of coming back to this idea of these like mutualisms versus parasitism, but also just that there's so much we don't know about all of these things. And so yes, there's been a lot of work. I'm sure somebody up here would argue needs to be more on human microbiomes, but I like to think about, the other 6,500 species of mammals and how little we know about their microbiomes, because we can't always take what we know about humans and put it into wild populations, or take what we know from wild populations and put it into humans, because we live in very different environments and there's a different background to how we are obtaining microbes and how we're interacting with our parasites. And it doesn't just vary from like species to species, it varies from population to population in what bacteria and parasites are being exposed to. And I don't remember what the prompt was, but I'm just gonna say
it was exactly that.
The moral of the story is, let's do more science.
Absolutely. What a wonderful way to end. Thank you, Kayce. Wow, that was like a power 45. I can't believe the time went so quickly. I think we have time perhaps for maybe 2 questions, and then afterwards, a little bit different this year, there's going to be a bit of a mingle, meet and greet out in the foyer, so you can keep asking your questions after the discussion. But the Natural History Museum staff will come around with a microphone. If you raise your hand, they'll hold the mic while you ask a question.
Yes, first of all, thank you very much for attending this. This was wonderful. In the last 5 to 10 years, I would say, I think it's been increasingly seen in society, the introduction of microplastics and finding that. How is that affecting microbiomes as you're seeing, both, as we're talking about dirt/soil, or are we talking about our own biologies and stuff like that? And is there possibly, in your opinion, a solution that might come from the study of our microbiology and microbes?
I have profound opinions.
It's like a game, like connect the thing to the microbiome.
So we've done studies in our coastal environments up and down. In San Diego, we have a hopefully thriving shellfish industry, oysters, and up here just north of LA, we have abalone. And a lot of these are filter feeders, right? They pick up on whatever particles they can find and they munch them up. And we do find extremely high levels of microplastics in these animals. We have not yet found any severe health implications for either the oysters, abalone, or the humans eating them due to those exposures.
I'm shocked.
Yeah, I know everybody's like, oh, well, surely they must be killing us. We can't find anything. I can put monstrously high levels of some of these microplastic contaminants in rodent populations, and they don't seem to react or respond. It seems like it's pretty inert, which I know is a horribly unpopular opinion, probably in this audience.
An anticlimax.
Please don't hurt me. But the one thing we have found, just to bring it back to happiness, or the old destruction, those act as a little carrier for bacterial biofilms to grow. And as our oceans warm, and our oceans, you'll want to, as a marine oceanographer that doesn't study marine oceanography, I can tell you this, the oceans have absorbed the same amount of energy as 20 nuclear reactions going off every single second for the last 25 years. That's how much energy the top 100 meters of the ocean is stored. And it's reached critical limits. Within 4 years, we are going to hit complete saturation of the top 100 meters. You want a disaster? That's a disaster waiting to happen. Then the microplastics won't matter. But it does relate to microplastics. Sorry, ADHD again. I really want to see what ADHD is in sign language. That's it, yeah. That's me. The bad bugs that make us sick are becoming more prevalent. Vibrio parahemolyticus. This is you get a dodgy oyster, it's probably got Vibrio parahemolyticus in it. And that grows really well on microplastics in the ocean. And what we found is that oysters that have microplastics covered in Vibrio parahemolyticus, when they consume them, that tends to contaminate the oyster's tissues more than normal bugs. So we believe that the microplastics may be acting as a way of transmitting these pathogens into there, and we might see an increased level of toxic oysters. You can get around that, though. For anyone in the oyster industry, I'm a big promoter of the industry.
Yeah,
you just got to clean out the oysters more. We go through a process called depulperation, where we wash them out with sterile water, and that's what you get most of the time when you go into a fresh situation. But you don't pick the oysters out of the water and just eat them raw, or be very, very careful, or have a toilet nearby, and a hospital with some antibiotics. That'd be good.
That was a great question and a great answer.
I think we can.
I think we can move on to, having our continuing our questions outside. Before we go, though, let's give a massive round of applause to our guests, Dr. Bell, Dr. Gilbert. That was an incredible conversation. So we're going to have our mingle outside. It'll just be in the foyer, but don't forget to check out the main stage in the North American Mammal Hall on level 1. At 8 p.m. is Georgia Mack, and finally at 9 p.m. is this evening's headliner, JSOM. So don't miss the live music as well and all the other activations happening around the museum. Thank you for joining our discussion. Thanks again.